Hello. So today we're going to present about this paper called Genetic Analysis of Early Onset Alzheimer's Disease Using Next Generation Sequencing. This paper was originally published on Nature Scientific Reports in June 2019 by a team from Korea. And this is our outline. First, I'm going to introduce some basic and background knowledge about Alzheimer's disease and other neurodegenerative diseases. And next, I'm going to talk about the next generation sequencing methods, which is ion torrent PGM used in this article, and about results, discussion, and conclusion. So let's start with the introduction. Alzheimer's disease is the main reason behind most dementia cases out there, which is 50 to 75% of them. While it typically strikes later in life, it can also show up unnaturally early in some people before age 65. We call that early onset Alzheimer's disease, or EOAD. For this early form, scientists have managed to pin down just 3 genes, APP, PSEN1, and PSEN2 as the main causative factors. However, there are far more mutations. Over 270 of them have been found in these genes that can basically hardwire someone to develop inherited Alzheimer's disease that runs in families. But here's where it gets hard for scientists and doctors. Those same genetic glitches that predispose someone to early Alzheimer's can somehow also lead to other neurodegenerative diseases like Parkinson's or frontotemporal dementia in some cases. On the other hand, the common late onset Alzheimer's that show up after 65 is a whole different genetic story. Over 20 genes have been linked to it so far, each adding just a little extra risk rather than causing it outright. To make matters even more confusing, various brain diseases like Alzheimer's, Parkinson's, and prion diseases can actually look really similar symptom-wise, making it tough for doctors to tell them apart clinically. This crossover suggests some shady underlying links between their genes and pathways. So to try to crack these genetic puzzles, researchers are using NGS to deeply analyze the genes of early Alzheimer's patients without any obvious culprits. Mapping out the full genetic landscape is critical for better diagnosing, understanding what goes haywire, and ultimately hoping to develop better treatments to take down this serious condition that impacts millions worldwide. Next, I'm going to introduce the main NGS methods that is used in this study, but first, let's look at a video.
Next-generation sequencing is a powerful technology which can interrogate many targets at the same time, from just a few genes to all of the individual nucleotides in a whole genome. Ion torrent technology from Thermo Fisher Scientific takes an entirely new approach to next-generation sequencing, making it massively scalable, faster, simpler, and more affordable than ever before. The sequencing of DNA is done using a semiconductor chip, similar to the chip found in your digital camera. While the chip in your digital camera has a sensing layer covered with millions of pixels that translate light into digital information, an ion chip has millions of wells covering those pixels. These wells capture chemical information from DNA sequencing and translate it into digital information, or base calls. The sequencing process starts when a sample of DNA is cut up into millions of fragments. Each fragment then attaches to its own bead and is copied until it covers the bead. This automated process covers millions of beads with millions of different fragments. These beads then flow across the chip, each depositing into a well. Then the chip is flooded with one of the 4 DNA nucleotides. Whenever a nucleotide is incorporated into a single strand of DNA, a hydrogen ion is released. This is how the ion torrent system sequences DNA. The release of the hydrogen ion changes the pH of the solution in the well. An ion-sensitive layer beneath the well measures that change in pH and converts it to voltage. This voltage change is recorded, indicating that the nucleotide was incorporated and a base was called. In essence, each well works as the world's smallest pH meter. The process is repeated every 15 seconds with a different nucleotide washing over the chip. For example, cytosine. A polymerase incorporates the C nucleotide in the DNA strand if a complementary G molecule is present. If the nucleotide is not complementary to the next base, no ion is released, no voltage change is recorded, and no base is called. If there are two identical bases next to each other, two nucleotides are incorporated, the voltage doubles, and the chip will record two identical bases called. This process happens simultaneously in millions of wells. That's why it's often described as massive parallel sequencing. The ion chips help you scale the workflow to your research needs so that you can run both small and large-scale projects without the need to change platforms. And the semiconductor approach helps you implement a significantly faster NGS workflow. There are many possible research applications with NGS, from whole genome sequencing to identify association of genomic variation with different diseases, to targeted NGS, where the whole power of NGS is used to interrogate a defined number of targets with possible significance in the pathogenesis. Ion torrent technology from Thermo Fisher Scientific.
So the Ion Torrent PGM is a next-generation sequencing platform that uses a semiconductor-based technology for DNA sequencing. It works based on the principle of detecting the released hydrogen ions during the incorporation of nucleotides during DNA synthesis. And here's a simplified explanation of how the Ion Torrent PGM works. So first is the library preparation. The DNA sample is first fragmented into smaller pieces, and adapters are ligated to the ends of these fragments. This step prepares the DNA library for sequencing. Number two is clonal amplification. The DNA library is then amplified using an emulsion PCR process. This step creates millions of clonal copies of each fragment on the surface of microscopic beads. Number three is loading the chip. The amplified DNA-coated beads are loaded onto a semiconductor chip, which contains millions of tiny wells. Each well is designed to accommodate a single bead. Number four is sequencing reaction. The chip is flooded with one nucleotide, A, C, G, or T, at a time. When a nucleotide is incorporated into the growing DNA strand during the sequencing reaction, a hydrogen ion is released. Number five is ion detection. The ion sensor at the bottom of each well detects the release of hydrogen ions. The more nucleotides are incorporated, the higher the signal from the release of hydrogen ions. Number six is signal processing. The ion sensor converts the chemical signal into a digital signal, which is then processed and translated into nucleotide sequences. Number seven is base calling. The sequence of nucleotides is determined based on the intensity of the signals from each well, allowing the reconstruction of the original DNA sequence. The Ion Torrent PGM sequencing technology is known for its rapid sequencing speed, scalability, and relatively low cost compared to other NGS platforms. However, it generally produces shorter read lengths compared to some other NGS technologies, which can limit its application in certain areas, such as de novo genome assembly or the detection of structural variation. And this section of the results talks about several mutations found in the PSEN1 and PSEN2 genes, which are linked to the early onset Alzheimer's disease and other neurological conditions. Here's an explanation of the mutations mentioned. So the first one is the PSEN1 gene mutations, and the first of its identified is the threonine 119 isoleucine mutation. This is found in a 64-year-old patient with early onset Alzheimer's. This mutation is located in an important region and may have similar effects as another known harmful mutation. Number two is glycine 209 alanine mutation. This is a mutation found in a 54-year-old woman with progressive memory loss. And number three is a glycine 417 alanine mutation. This is found in a 37-year-old man with symptoms of both Alzheimer's and Parkinson's disease. This mutation may cause problems in the protein structure and affect the gene's function. And for PSEN2 mutations, this is a histidine 169 asparagine mutation, which is found in a 63-year-old woman with early onset Alzheimer's. This mutation was previously seen in two Chinese patients, one with late onset Alzheimer's and the other with frontotemporal dementia. In summary, this describes several new and previously known mutations in the PSEN1 and PSEN2 genes that were identified in patients with various forms of dementia and neurological conditions, such as early onset Alzheimer's, Parkinson's-like symptoms, and memory problems. It provides detail about the potential effects of these mutations based on their locations within the proteins and their similarities to other harmful mutations. And this is the figure of detection of PSEN1 and PSEN2 genes mutation in early onset Alzheimer's disease. For the top left is the A figure, and which means the DNA sequencing data of the 4 mutations found in this study. And B is the location of PSEN1 and PSEN2 mutations in the genomic DNAs and protein. And C is the possible protein structure changes associated with PSEN1 and PSEN2 mutations. And this part of the results suggests that the study identified 67 non-synonymous variants in genes associated with increased risk for late-onset Alzheimer's disease, and 10 missense variants were detected in the SOR1 gene, including a common variant present in 33% of patients that increased A-beta-42 secretion. 24 missense variants were found in ABCA7, some predicted to be potentially damaging based on in silico analysis. And the study identified 17 novel and missense variants in 21 genes associated with other neurodegenerative diseases like frontotemporal dementia, Parkinson's, prion diseases, and others, suggesting potential genetic overlap between these conditions and EOAD. Network analysis revealed the identified genes may play roles in amyloid beta metabolism, formation, and APP processing, as well as oxidative stress response, synaptic transmission, and other AD-related pathways. The findings suggest screening a broader panel of neurodegenerative disease genes in EOAD patients could provide insights into the missing heritability and disease mechanisms, given the genetic overlap observed. Expanding the screening beyond known EOAD genes is warranted. And the key findings include: the first one is the SOR1. 10 missense variants were identified, with approximately 33% of patients carrying a common variant known to increase A-beta-42 secretions. Number 2 is the ABCA7. 24 missense variants were found, including a stop-gain mutation and a significant frame-shift mutation, both suggesting loss of function mechanisms related to AD. And number 3 is the CLU. A novel variant was identified in EOAD patients, highlighting its potential role in AD pathogenesis. The study supports the involvement of these gene variants in AD and suggests they may explain the genetic etiology of EOAD in the cohort.
Next, I will be discussing the mutations in neurodegeneration-associated genes identified in patients with EOAD as presented in the study. Firstly, it's important to note that AD shares overlapping clinical and neuropathological features with other neurodegenerative dimensions, such as FTD, CBD, PSP, and CJD. This overlap can lead to misdiagnosis in approximately 70 to 30% of AD cases. Despite the identification of rare high-parent mutation in APP, PSEN1, and PSEN2, these mutations account for only a small fraction of EOAD families, leaving many autosomal dominant pedigrees genetically unexplained. So, they use NGS to identify 70 non-synonymous variants in 21 dimensions-related genes in the patients, and focus on the PRNP and GRN gene. First, mutations in the PRNP gene were detected at a high frequency in the study. Approximately 3.3% of AD patients show heterozygosity in codon 129, while codon 219 was detected in 10.9% of cases. These common variants, although potentially benign polymorphism, could also be risk factors for CJD. Next, significant pathological overlap was observed between FTD and AD for 2 GRN mutations, serine 40S and arginine 564-16 mutation. Although previously reported with unclear function, has been suggested to be associated with AD. These findings indicate that several mutations in GRN may be risk factors for AD, highlighting possible pathological and genetic overlaps between these disorders. And these findings suggest that the genetics of AD may be more complex and heterogeneous than previously thought. Many potential variants are present in genes associated with other forms of neurodegeneration, such as PD, FTD, ALS, and prion disease. I will now discuss the in silico gene-protein functional interaction network constructed in this study to better understand the molecular interaction involved in EOAD. Due to incomplete understanding of the molecular mechanism, the researchers aim to unravel the complex interplay between various genes and proteins associated with EOAD. They employ a tool called Kruger. Kruger is a plugin of Cytoscape, which is an open-source bioinformatics software platform for visualizing molecular interaction networks. By using Kruger, researchers can visualize the non-reductant biological terms for large clusters of genes in a functionally grouped network. They conduct interaction network analysis to assess 35 gene-gene interaction which may be associated with AD. This is the analysis result, which reveals several key interaction clusters within the network. According to the gene marked by a circle in the figure, we can find that PSEN1 and PSEN2 may be involved in amyloid mechanisms such as amyloid beta formation and APP metabolism. Other genes potentially also play a direct or indirect role in amyloid metabolism, including SOIL1, ABCA7, CLU, SNCA, and LRRK2. There are also several genes that may be involved in other mechanisms, such as by regulating cellular responses to oxidative stress or synaptic transmission. This result may lead to improved therapeutics targeting with amyloid beta accumulation as the dominant factor. Next, we are going to talk about the discussion. The study demonstrated that mutations in AD-related genes including APP, PSEN1, and PSEN2 cause, contribute to, and modify the risks of AD. But the clinical and pathological overlap between AD and other neurodegenerative disease, demonstrating the variants contribute to disease at a genome-wide level, and frequency of rare mutation should be determined, as this variant may contribute to disease through different mechanisms. The main limitation is that the majority of EOAD cases remain genetically unexplained. In the study, less than 6% of patients with EOAD in the study carry PSEN1 gene. Meanwhile, the majority of EOAD cases and genes implicated in LOAD remains genetically unexplained. The second limitation is that some family members of patients refuse genetic testing, which makes researchers think can get more statistical genetic data. In contrast to sequencing data, the Kruger revealed complex interaction between PSEN1, PSEN2, and several additional AD risk genes. Additionally, Kruger interactions suggest that other neurodegenerative disease-causing genes impact amyloid-associated pathways such as LRRK2, PIK7, and SNCA. First is LRRK2. Its mutants were associated with reduced amyloid beta levels in patients with CSF or PD. Also, it was suggested as a potential risk factor for AD. Meanwhile, mutated LRRK2 protein may enhance the phosphorylation of intracellular domain of APP, resulting in neurotoxicity. Second is PIK7, which may impact AD onset, as their expression levels increased and gene interaction were observed in cases of oxidative stress. Oxidative stress has been suggested to play a significant role in neurodegeneration and the onset of different neurodegenerative disease. Next is SNCA, which may also impact AD onset, as the interaction of amyloid beta peptides and alpha-synuclein interaction may result in neurotoxicity through enhanced oxidative stress, impaired calcium metabolism, and abnormal mitochondrial pathways. That's briefly what we have looked at. The researcher team discovered three important things. First, the mutations in APP, PSEN1, and PSEN2 are relatively rare among patients with EOAD. Second, rare missense mutations were also found, which may contribute to neurodegeneration. And third, several mutations in AD risk factor genes. Based on the above description, these findings are helpful for additional genetic profiling of patients with AD and to improve diagnosis as well as broaden the knowledge of the possible genes and pathways associated with neurodegeneration. And about the future, they want to provide a foundation for personalized medicine, which may be accomplished through gene identification study or trials for biomarker selection or compound testing. Also, they want to get more data for further investigation about interaction and pathways that may increase the understanding of the pathway mechanism related to the mutation. These results may lead to improved therapeutics targeting with amyloid beta accumulation as the dominant factor. And finally is our extension question. How can the data from in silico gene and protein functional interaction network be utilized to predict novel therapeutic targets for EOAD? And next is how could future researchers integrate environmental and lifestyle factors with genetic data to provide a more comprehensive understanding of EOAD's disease etiology? And it is the end of our presentation. Thanks for listening.